Hello everyone, welcome back to my course on Microbial Biotechnology. We are in module 5 where we are discussing the control of microorganisms. Today we will be focusing on the chemical methods of microbial control. So in the last lecture we have discussed about the physical methods of microbial control. So today we will be discussing about the chemical methods of microbial control, under which we have various agents like aldehydes, phenol and its derivatives, and then we have alcohols. We also use certain halogens like fluorine, chlorine, iodine. Then we have certain metals which we use for disinfection, and then we have surfactants like soaps and detergents, and then hydrogen peroxide, peracetic acid, and alkylating agents like ethylene oxide and beta propionolactone. In aldehydes, we have various compounds like formaldehyde, glutaraldehyde, etc. So, we will be discussing these in detail one by one. Before that, let us have an overview of the chemical methods of microbial control. So, chemical control of microorganisms refer to the use of various chemical substances which we have discussed in the earlier list, or compounds which prevent, inhibit or eliminate growth and spread of microorganisms. Various chemicals exhibit varying degree of effectiveness against specific types of microorganisms. Chemical agents used for this purpose are known as antimicrobial agents or disinfectants. These substances can function by disrupting the cell walls or membranes of microorganisms, interfering with their metabolism or damaging their genetic material. Antimicrobial chemicals are often utilized in various settings such as healthcare facilities, research laboratories, food processing industries, and household to maintain hygiene, prevent infections, and control the spread of disease. So, in this lecture, briefly we will be discussing about some of the terminologies we learned in the last lecture and the various chemical agents which we have already briefed in the table earlier. So, let us briefly have a recap of the terminology that we learned in the last class. So, sterilization is the process of destroying all living microorganisms and viruses. Disinfection is the elimination of microorganisms from inanimate objects or surfaces. Decontamination is the treatment of an object or inanimate surface to make it safe to handle. Disinfectant are agents used to disinfect inanimate objects but generally toxic to use on human tissues. Antiseptic is an agent that kills or inhibits growth of microbes but is safe to use on human tissue. Sanitary is an agent that reduces but may not eliminate microbial numbers to a safe level, and it can be used on humans or human tissue. Cidal are basically an agent which will kill microorganisms and viruses, so we will call them as fungicidal or bactericidal. And then we have certain agents which I will call as static agents. These will inhibit the growth of microorganisms like bacteriostatic or fungistatic. Now, all these chemicals that we are going to discuss today have certain properties which make them good chemical control agents. So, what are these properties? Number one, the chemicals should show a broad spectrum of antimicrobial activity even at low concentrations. For effective use, it should be soluble in water or other commonly used solvents if not water. These agents should have the capacity to effectively penetrate biological membranes for germicidal action. In a solution, the chemicals must be stable and should not show loss in germicidal action. Chemicals should be selectively lethal towards the microbes and should not be toxic to humans and animals, but lethal to microbes. Chemicals should be non-corroding and non-staining. These chemicals that are widely available must be widely available and are cost effective, are the most preferred. Now, let us discuss one of these chemicals, the phenol, which is also very common in our household use. So, this is a known carboxylic acid, and it is one of the earliest antiseptic substances. Here you can see a vial of carboxylic acid, then you can see medallion commemorating Joseph Lister's development of carboxylic spray for antiseptic use during surgery. Phenol exhibits bacteriostatic properties within concentration range of around 0.1 to 1% and demonstrates bactericidal and fungicidal effects at 1 to 2%. So, the concentration of phenol will actually also dictate its property, whether it is going to be static or it is going to be cidal. So, as already discussed, Joseph Lister pioneered antiseptic surgery by introducing carboxylic acid to reduce infections during operations in the late 19th century. The mechanism of phenol as a disinfectant involves its ability to disrupt and denature proteins, leading to the inactivation of microorganisms. So, you have here various structures of some widely used phenol derivatives, and you can see metacresol, then orthocresol, and also triclosan over here. What are the mechanism of action? As already briefly discussed, phenol can interact with membrane proteins, disrupt the integrity of the cell membranes, and this leads to increased permeability and cellular leakage. Phenol also denature proteins within the microbial cells, leading to interference in normal functions including enzymatic activities. Phenol can also induce oxidative stress within microbial cells by generating reactive oxygen species. This oxidative damage can lead to the degradation of cellular components and contribute to the overall antimicrobial effect. So, these are some of the phenol derivatives like the cresols, including orthocresols, metacresols and paracresols, which are effective microbial agents. They are widely used in the disinfectant industry, antiseptics and industrial settings for the broad spectrum microbial activity. So, this is for example a formulation with triclosan, and then roughly around 0.75%. Then this triclosan, as we are discussing, is a chlorinated aromatic compound with broad spectrum antimicrobial properties. It inhibits bacterial fatty acid synthesis, disrupting cell membranes, and is commonly used in consumer products like soaps, toothpastes and deodorants as an antibacterial agent. So, another phenol derivative is chloroxylenol, for example, which is marketed as Dettol, and a widely used brand of antiseptic and disinfectant with these particular compound chloroxylenol as its active ingredient. Then we have thymol, which is produced by the alkylation of metacresol and propene, disrupts cell membrane and exhibits antioxidant activities. These are found in some mouthwashes like Listerine, and it is a disinfectant and natural antimicrobial formulations. The next class of compounds or agents that are used as disinfectants are alcohols. Alcohols are inexpensive and easily available. These are widely used in disinfecting laboratory and chemical instruments and as hand sanitizers. The germicidal effect of alcohol increases with increase in molecular weight. Methanol is the least germicidal. The effect increases in order for ethanol, propanol, butanol and so on. Ethanol and propanol are mostly used alcohols as disinfectant agents. For example, ethanol concentrations of around 60 to 80% also have a virucidal effect against lipophilic viruses and many hydrophilic viruses. So, here you can see two brands of hand sanitizers. On the left it has isopropyl based formulation. Then what is the mechanism of action of alcohol in controlling microbes? Alcohol in appropriate concentrations works by first solubilizing the lipid bilayer of cell wall in the gram negative cells and membranes to create pores. Alcohol then enters the cytoplasm through these pores to denature the cellular proteins and finally killing the bacteria. It must be mentioned that 70% alcohol is more effective than absolute alcohol. This is because absolute alcohol brings about extreme dehydration, causing the cells to shrink, thus preventing the entry of alcohol into the cell. This leads to bacteriostatic action, but not bactericidal. At 70% concentration, both dehydrating and denaturing effects work almost in equilibrium, causing bactericidal effects. One major disadvantage of using alcohol is its volatility, which limits its effectiveness to immediate application. Let us now discuss about the halogens, for example, fluorine. And then you have these fluoride varnish by Colgate, used by dentist in this toothpaste over here. So, due to their high reactivity, halogens are often used as antiseptics and disinfectants. Fluorine, for instance, is widely used in toothpaste formulations as the main active ingredient, since it contributes to the prevention of dental caries. While fluoride mainly functions by its incorporation into tooth enamel, making it more resistant to corrosion, fluoride also has bacteriostatic actions. It interferes with the metabolism of plaque forming bacteria and reducing their production and the acids that contribute to decay. Chlorine is another halogen which is widely used among the halogens overall, especially as sodium hypochlorite as a household blaze solution and calcium hypochlorite as bleaching powder. This is inexpensive and very fast acting, has a broad spectrum of microbicidal activities. Chlorine products may cause ocular irritation, of course, and gastric burns. It can corrode metals and can cause bleaching of fabrics. Inorganic chlorine solution is used for spot disinfection of countertops and floors, for decontaminating blood spills, and as a disinfectant for laundry and dental applications. Chlorine in the form of monochlorine, monochloramine, chlorine dioxide or hypochlorites has been widely used for a long time in treatment of municipal water. Now, what is the mechanism of action of chlorine? The exact mechanism by which free chlorine causes disinfection is not fully understood, but it can be attributed to a number of factors like oxidation of sulfur hydryl enzymes and amino acids, ring chlorination of amino acids, loss of intracellular contents, decreased uptake of nutrients, inhibition of protein synthesis, decreased oxygen uptake, oxidation of respiratory components, decreased adenosine triphosphate production which breaks in DNA, then depressed DNA synthesis. The actual microbiocidal mechanism of chlorine might involve a combination of many of these factors. Low concentrations of freely available chlorine have a biocidal effect on mycoplasma and vegetative bacteria. Higher concentrations of chlorine are required to kill M. tuberculosis or Mycobacterium tuberculosis. Studies have reported inactivation of 25 different viruses in 10 minutes with 1:100 dilution, including HIV. Another halogen that is used as a disinfectant agent is iodine. An iodophore is a mixture of iodine with a solubilizing agent or carrier, which releases small amounts of free iodine in aqueous solution used as antiseptic agents. So, you can see here the structure of povidone iodine complex in this particular figure, which is being used as a disinfectant in medical clinics. The most widely used iodophore, as we have already discussed, the povidone ion is a mixture of polyvinyl pyrrolidone with iodine. Free iodine can penetrate the cell wall of microorganisms quickly, and lethal effects are believed to result from disruption of protein and nucleic acid structure and synthesis. Besides the use as an antiseptic, iodophores have been used for disinfecting blood culture bottles and medical equipments. Let us now discuss about certain metals which are used as chemical control agents. Metals have been used as disinfectants for a very long time, but their uses have now been limited due to their toxicity. Metal binds to sulfur containing amino acids of cellular proteins and can inhibit enzymatic activity. Let us discuss about copper. Copper sulfate is a common algicide used to control algal growth in swimming pools and fish tanks. Then we have zinc. For example, zinc chloride is an ingredient for mouthwashes, while zinc oxide is found in a variety of products including topical antiseptic creams such as calamine lotion, diaper ointments, baby powder and dandruff shampoos. Then we have, of course, toxic metals like mercury. It was once used widely to treat syphilis, and tinctures like mercurochrome and marthiolate were used as antiseptics. So, here you can see a vial of mercurochrome on this picture, and you can see the compound in this diagram. Mercury, however, is not selectively toxic to microbial cells and may bioaccumulate in human or animal cells. In excess amounts, mercury is toxic to the central nervous, digestive and renal systems at high concentration and can cause bioaccumulation in fish. So, another metal that is used as chemical control agent is silver. Silver sulfadiazine, which is commercially sold as silvadine cream, is widely used for treatment, for example, of topical wounds, especially for prevention of infection in burn wounds. Silver nitrate drops were used for prevention of ophthalmia, neonatorum, eye infections that can occur due to exposure to pathogens in the birth canal in newborns. Let us now discuss about another class of compounds, the aldehydes. Let us begin with formaldehyde, which in its aqueous form, principally in 37% weight by volume formulation, is used as a disinfectant, as the solution has bactericidal, tuberculocidal, fungicidal, virucidal and sporicidal effects. Here you can see a fish specimen preserved in formalin in a museum, and so it can increase the shelf life by many years and decades. Formaldehyde is used in vaccine manufacturing for inactivation of toxins and pathogens, and as a biocidal agent in the production of cosmetics and animal feeds due to its antimicrobial properties. Formaldehyde is also used in preserving anatomical specimen and for embalming corpses and carcasses. Formaldehyde, however, is classified as a carcinogen. Ingestion can be lethal, and limited exposure can cause respiratory and skin irritation. What is the mechanism of action of formaldehyde? So, it brings about the monohydroxymethylation of adenine on both DNA and RNA, which blocks genome reading. Formaldehyde can also cause monohydroxymethylation of the N-terminal amino acid residue or amino acids containing nitrogen or sulfur in their side chain by reacting with the amino or sulfhydryl groups. Such reactions of monohydroxymethylations result in formation of skiff bases which can cross-link with arginine and tyrosine, and to a lesser extent with glutamine, asparagine, tryptophan and histidine residues. These cross-links form inter and intramolecular methylene bridges, causes the proteins to coagulate. Another class of aldehyde example is glutaraldehyde, which is a saturated dialdehyde, widely accepted as a strong disinfectant and sterilant. While aqueous solutions of glutaraldehyde are acidic and lack biocidal activity, activation by changing the pH to 7.5 to 8.5% makes glutaraldehyde a potent disinfectant. Glutaraldehyde is non-corrosive to metal, plastic, lanced equipments and rubber, and is widely used for disinfecting medical equipments such as endoscopes, spirometry tubing, dialyzers, transducers and devices for anesthesia, respiratory therapy and hemodialysis. However, prolonged exposure can lead to skin irritation, dermatitis, mucous membrane irritation and pulmonary symptoms. Reported effects in healthcare workers include epistaxis, allergic contact, dermatitis, asthma and rhinitis. What is the mechanism action of glutaraldehyde? So, you can see here glutaraldehyde undergoing polymerization to form polyglutaraldehyde, and it leads to cross-linking with microbial and protein the peptides, and then you can see here the reaction in the gram negative in the outer membrane and in the gram positive in the peptidoglycan. So, briefly, the biocidal activity of glutaraldehyde also results from its alkylation of sulfahydryl, hydroxyl, carboxyl and amino groups in the RNA, DNA and protein of microorganisms. The mechanisms of nucleic acid alkylation is the same as that for formaldehyde. The alkylation of proteins occurs through aldol condensation followed by elimination of water. Several amino acids can then link onto glutaraldehyde leading to formation of bridges. Another type of aldehyde is the orthotelaldehyde or OPA. Like glutaraldehyde, orthotelaldehyde also interacts with nucleic acids, amino acids and proteins of microorganisms. However, compared to glutaraldehyde, OPA is a less potent cross-linking agent, but has numerous advantages over glutaraldehyde. OPA shows excellent stability over a wide range of pH ranging from 3 to 9. OPA does not cause irritation to the eyes and nasal passages and has a barely perceptible odor and does not require exposure monitoring. Lipophilic aromatic nature of OPA can assist its uptake through the outer layers of microbacteria and gram-negative bacteria. OPA shows very strong bioactivity against glutaraldehyde resistant mycobacteria and a potent sporicidal effect against Bacillus atrophaeus spores. A potential disadvantage of OPA is that it stains proteins gray, including unprotected skin, and thus must be handled with caution. Let us now discuss about another class of chemicals which are used in controlling microbes, the alkylating agents like ethylene dioxide. It is an alkylating agent with high penetration used for gas, phase, sterilization of medical equipments and instruments, packaging material and clothing, surgical and scientific equipments. So, here in this figure we can see the alkylating action of ethylene dioxide, alkylation of sulfahydryl group on the top and then the alkylation of guanine in below figure B. Ethylene oxides can cause alkylation of both the proteins and nucleic acids attacking the sulfahydryl, amino or hydroxyl groups. These alkylations interfere with normal cellular metabolism and replication. Ethylene oxide, however, must be handled with great care owing to its explosive and carcinogenic nature. Let us now discuss about another alkylating agent which is briefly known as BPL or beta propionyl. BPL is widely known for its virucidal and sporicidal effects. It is used in vaccines for virus inactivation, but its usage as growth control is limited due to its carcinogenicity. You can see here the skeletal formula of BPL in this picture. BPL shows potent virucidal effect against several pathogens like equine encephalitis, rabies, poliomyelitis, Coxsackie, herpes simplex virus and measles, hog cholera, influenza, hepatitis and even E. coli and tetraphase. What is the mechanism of action of BPL or beta propionyl? BPL primarily brings about alkylation of guanine bases at the N7 atom and to a lesser extent the N1 atom of guanine and N3 atom of adenine bases, subsequently leading to GC80 transition mutations or can cause cross-linking of the double helix. And you can see here in this picture the alkylating actions of BPL. In figure A you can see the alkylation of deoxyguanosine. In B you can see the alkylation of deoxyadenosine. Let us now discuss about another class of chemical agents which control microbial growth, which we briefly call as surfactants. These are surface active agents and are a group of chemical compounds that lower the surface tension of water. Surfactants form the major components in soaps and detergents. Most surfactants do not effectively destroy microorganisms, but act by mechanically removing the organisms from the surface. An important class of surfactant, however, called quaternary ammonium compounds exhibit antimicrobial properties. Let us discuss about the soaps. So, we regularly use soaps even if you are not a clinician or a researcher in a biological lab. This type of agent is known to you. So, here this is an advertisement or awareness program, for example, by World Health Organization, which tells about protecting yourself and others from getting sick by just washing your hands regularly after coughing or sneezing when caring for the sick, because the sick person may have certain contamination. Before, during and after you prepare food, before eating, after toilet use, when hands are visibly dirty, after handling animals or animal waste, and then you can use soaps and surfactants in this entire cleaning process. Soaps are salts of long chain fatty acids and have both polar and nonpolar regions, allowing them to interact with polar and nonpolar regions in other molecules. Soaps do not kill or inhibit microbial growth, and so are not considered antiseptics or disinfectants. However, they can interact with nonpolar oils and grease to create emulsions in water, loosening and lifting away dirt and microbes from surfaces and skin. Some soaps contain added bacteriostatic agents such as trichlorocarbon or chlorofluorocarbon that introduces antiseptic or disinfectant properties to the soaps. Another surfactant are the detergent. Detergent is a surfactant or a mixture of surfactant molecules with cleansing properties when in dilute form. Based on their charges, detergents may be classified as anionic detergent. These are widely used for laundry and can mechanically remove microorganisms. Examples include sodium alkyl sulfates such as sodium lauryl sulfate and sodium alkyl benzene sulfonates such as sodium 4,1-dodecyl benzene sulfonate. Then we have the cationic detergents, which include an important class of disinfectants and antiseptics called the quaternary ammonium salts. And for example, we have Senja, alconium chlorides and acetyl pyrimidine chloride. Then we have non-ionic detergents, which are neither cationic or anionic, like polyoxyethylene or glycoside based detergents. Some of the examples include Twin, Triton and the Breeze series. Then we have deuteronic detergents. These exist as deuterions within a particular pH range. Examples include CHAPS. Let us now discuss about the quaternary ammonium products, or briefly which are called as quats. So, chemically these quaternary ammonium compounds are organically substituted ammonium compounds in which 4 of the substituent radicals R1 to R4, as you can see in this image, are alkyl or heterocyclic radicals, and there is a 5th X is a halide, sulfate or similar radical. Each quaternary ammonium fluoride exhibits antimicrobial characteristics and are widely used in medical fields to disinfect patient care supplies or equipment such as cystoscopes or cardiac catheters. Some of the quaternary ammonium compounds used in healthcare are alkyl dimethyl benzyl ammonium chloride, alkyl didecyl dimethyl ammonium chloride and dialkyl dimethyl ammonium chloride. What is the mechanism action of quats? The quats share certain physical and chemical properties with phospholipids and have hydrophilic and hydrophobic ends. As such, quats can insert into the bacterial phospholipid bilayer and disrupt membrane integrity. So, you can see here the mechanism action of this quaternary ammonium compound, which is disrupting the membrane integrity. The cationic charges of quats have been associated with the antimicrobial properties as neutralizing results in loss of antimicrobial function. Quats used as hospital disinfectants have been known to possess fungicidal and bactericidal activities and have virucidal activity against lipophilic viruses. However, these lack sporicidal and tuberculocidal activities and are ineffective against hydrophilic non-enveloped viruses. Another compound that is used as chemical control agent is the hydrogen peroxide, which shows antimicrobial activity against a wide range of microorganisms, including bacteria, yeast, fungi, viruses, and spores. So, here you can see hydrogen peroxide based household disinfectant. It works by producing destructive hydroxyl free radicals that can attack membrane, lipids, DNA, and other essential cell components at a concentration of around 0.5%. It demonstrates bactericidal and virucidal activity in 1 minute, and mycobactericidal and fungicidal activity in 5 minutes. Notably, most aerobic organisms and facultative anaerobes possesses the enzyme catalase, which protects cells from metabolically produced hydrogen peroxide by degrading hydrogen peroxide to water and oxygen. Let us now discuss about pyracetic acid. Pyracetic acid or peroxyacetic acid is an oxidating agent widely used as an disinfectant, and you can see the skeletal formula of pyracetic acid in this diagram. It has the ability to completely destroy microorganisms without any trace of organic matter and harmful byproducts. Its mechanism of action is believed to be involved being protein denaturation, disruption of cell wall permeability, and oxidation of sulfur hydryl and sulfur bonds in proteins and enzymes, although the exact mechanism remains a mystery. Pyracetic acid can bring about removal of microorganism even at very low concentrations, and also shows sporicidal and virucidal effects. So, with this we come to end of this discussion on chemical control agents. So, thank you for your patient hearing.